Thank you very much. So I would like to present to you the results from our exploratory phase 2 study in SLE with SM101. These are my disclaimers. Okay, just for SM101, a short description. So SM101 is the extracellular 2B part of the FCGIIB receptor. You can see here a schematic picture of SM101. The proposed mode of action is that it binds to autoimmune complexes on the FC part and therefore inhibits the binding to effector cells, FC receptors on effector cells. And here's just a little cartoon. You see SM101 in blue attached here to the autoimmune complexes on the FC part and then inhibits the further binding to activating FC gamma receptors overall on effector cells. And as you know, in SLE, there is an upregulation of activating FC receptors in comparison to the inhibiting 2B receptor. The clearance mechanism is postulated that it will be eliminated via the neonatal FC receptors, the autoimmune complexes, and therefore inhibits the further organ damage through the autoimmune complexes. So we have performed a phase 2 double-blind randomized clinical trial in patients with SLE. This was a 6-month study. The plan was to include 50 stable SLE patients with activity in terms of a Selena-Slater score of at least 6, and also serologically active patients with either a high anti-double-stranded DNA or a low complement C3 or both. We had also a small subset of patients who had a history of lupus nephritis, which was defined as a biopsy-proven glomerulonephritis within 5 years prior to the first IMP infusion and a mild proteinuria of at least 0.2 gram per day. The treatment was 6 and 12 mg per kg SM101 or placebo in a randomization scheme of 2 to 2 to 1, given weekly for 4 weeks. And this is important just to mention because after the 4 weeks of initial treatment, there was no further treatment administered to the patients. Concomitant SLE treatment was allowed, mainly, of course, corticosteroids, but it has to be stable for at least 4 weeks. And for other concomitant medications, then longer. Patients might have, in addition, also azathioprine or MMF with predefined limits to the dosage. And as for almost all SLE studies, the primary endpoint here in this early stage was safety, but also we had clinical scores like Slater bar, like PGA, and we have here also global responders, but this is basically the same as an SRI-4 responder index. Okay, this is the enrollment flow chart. Altogether, we had 22 active sites in Europe and also in Australia. Altogether, we had 51 patients who received at least 1 dose of treatment. And you see here down that how the distribution across the treatment group was. As I said, a small subset of patients had the definition of lupus nephritis, but this was a small subset of 14 patients overall. If you look at the patient baseline characteristics, it is very well distributed across treatment groups. If you look at the BILAG score, there appears to be that the 6 mg per kg patients had a slightly higher BILAG score at baseline, but the SLE duration, the Slater score, and the PGA score was very much the same at baseline. Almost all patients had a high anti-double-stranded DNA, of course, and in addition to that, also low complement C3. Almost all patients had concomitant steroids, around 90% of all of the treatment groups, and then in addition, MMF or azathioprine. There was one protocol deviator who received at baseline cyclophosphamide. Okay, just in terms of rescue medication, the mean is around 1/4 of the patients have received in the SM101 group rescue medication. This was defined as any increase of concomitant medication, immunosuppressive medication, or a new drug was given to the patient in terms of rescue medication. About 1/3 of the patients on placebo have received rescue medication during the treatment. Here you see just the differences in the mean scores from baseline. You see on the SLEDAR score there's quite a few fluctuation, and there appears to be not a big difference to month 6. For the BILAG score and for the PGA score, however, there appears to be a trend, at least for the 6 mg per kg and for the 12 mg per kg group seen here. It's fair to say that all of the parameters we have evaluated, the sample size was too small in order to show any significant difference, but at least you can see here a trend in the improvement of the score. If you now look at the SLEDAR responders, so we have looked at 4-point SLEDAR responder and up to 7-point SLEDAR responder, you see quite nicely that there is for the 4-point responder a trend in improvement with a higher dose. The orange bar here is always at 12 mg per kg dose group. Here 50% of the patients had a SLEDAR response, 4-point and higher, and this maintains throughout up to 7-point with 11% of the patients in the 12 mg per kg group. Overall, those patients didn't have a lot of BILAG flares, as you can see here. For example, during the study, the 12 mg per kg group overall, 11% of the patients had a BILAG A flare or 2B flares during the study. When you look what is behind the improvement in the BILAG score and SLEDAR score, this is mainly driven by an improvement in arthritis and in skin eruption. Sorry. And this is the same for the SLEDAR score, basically arthritis and rash. You see here, for example, for arthritis that for 88% in the placebo group compared to baseline remains just the same, where for improvement or remission was only associated with the SM101 dose groups. Remission here is a part of the improvement. So for remission, we had 36% of the patients in the 12 mg per kg group having a remission in arthritis by the end of the trial. This was a bit, let's say, different in the skin eruption, but also here only the SM101 treated patients had improvement or remission at the end of the study. PGA responder were basically the same. You see on the left-hand side that no PGA worsening, so PGA responders, if you like, 89% of the patients in the 12 mg per kg group, and the trend remained the same when you remove and those patients who had received rescue medication, so up to 67% had a response at week 24. So on this, just everybody knows, of course, about the SRI-4 index. Just we have done this as a post-hoc analysis, so basically this is the normal definition of an SRI-4 responders, SLEDAR score, a reduction of at least 4, no BILAG A or 2B flares during the trial, and no PGA worsening of 0.3 or higher during the clinical trial as well. And if you look now at this global responder or SRI-4 responder index here, you see there a nice trend, up to 39% of the patients on 12 mg per kg were responders. If you then remove those patients on rescue, it's still 39% in the 12 mg per kg group, so there appears to be no big difference. So this is quite promising. We then had to look at the lupus nephritis subset. These are really small numbers. As I told you, 14 patients only had the additional definition of lupus nephritis. Still, there is a nice, let's say, trend. Only those patients on treatment had a response, global response, and is maintained more or less the same if you remove those patients who have received rescue medication during the trial. Then important, of course, this was one of the main objectives of the study, is to evaluate the safety of SM101 in SLE patients. You see adverse events incidence of 10% or higher, and I would like just to point out the infections you see here. Again, 12 mg per kg is the orange bar. You have here the 6 mg per kg and you have here the 12 mg per kg is just the other way around, and here the placebo with in the orange bar, and then 6 mg per kg here. So there was no increased rate of infection, and overall there was also no dose-dependent increase in any of the adverse events we have seen in the study. And because infection is quite important, we have also done an overall analysis of the patient data we had so far. So SM101 has been also been investigated in an ITP clinical trial, in a small dose escalation clinical trial. You see here on the left-hand side, and you see here now in blue all the SM101 patients, and in gray is the placebo patients. So not surprisingly, the overall infection rate in SLE clinical trials is higher than in ITP clinical trials, but there is no increased infection risk for SM101. Of course, these are limited data up to 6 months of study period. The overall infection rate between placebo and SM101 treated patients was around 1/3. So in summary, this clinical trial, the phase 2 exploratory clinical trial, shows that there's an interesting efficacy signal for the clinical scores like SLEDAI, BALAC, and PGA. And it's important to mention that only 4 weekly infusions at the start were necessary in order to induce an improvement or trend in improvement in those scores at 6 months. This effect was most prominent in lupus nephritis subset in the few patients we have evaluated. Arthritis and rash, skin eruption were the major drivers for this improvement in the clinical scores. There were no specific SM101 adverse events seen in the overall population, and we have not detected any anti-drug antibody to SM101 in this clinical trial. And this results suggest that SM101 could be an efficacious and safe treatment in patients with SLE. I would like also add another thing, that we have of course evaluated cell subset changes and also anti-double-stranded DNA complement, anti-C1q, and other important parameters as well. But at the moment we couldn't identify any, let's say, single biomarker attributed really to the treatment effect of SM101, but we hope that we can get more insights in the future development of SM101 and SLE. My thanks goes to the investigator who have run the trial. Thank you very much for your attention.